Hello. Today, I am going to talk about a genetic engineering concept, which is gene expression regulation in prokaryotes. So, I am Amrita, an assistant professor in Krishna Jayanti University, and we are going to start the topic. To understand the gene expression regulation in prokaryotes, we must understand the concept of operons. Many prokaryotic genomes have a cluster of genes that are often transcribed together, and they are regulated also based on the environmental conditions. So today we are going to talk about two operons. One is a lac operon and one is the tryptophan operon. So the right side figure here shows a set of genes that are clustered at in one region, localized in the genome of a bacteria. And this operon takes care of the metabolic needs of the cells when there is presence of lactose rather than glucose in the environment. In general, your prokaryotes are also more inclined towards glucose for their metabolic needs. But let's say the glucose uh, amount is less in the environment and there is the presence of lactose. So cells have evolved a mechanism to utilize the lactose in place of glucose for their metabolic needs. So from this figure, we will understand that these genes that are clustered together have, each of these genes have different roles in the lactose metabolism. For example, when glucose is low, there is CAMP that is upregulated. This CAMP, cyclic AMP molecule, it binds to catabolite activator protein and that attaches to the cap site in this operon. Once this cap site is occupied, it promotes the RNA polymerase from attaching to the promoter region uh, and it transcribing the other genes. The rest of the genes other than operator here, LAC-Z, LAC-Y, LAC-A, codes for enzymes that metabolizes lactose for the energy requirement of the cell. So this is how one of the region in the operon is majorly utilized when the presence of glucose, that is the major metabolic component, is low. So this is the how this particular operon is actually regulated by one of this gene. Now there is one more regulation to this operon, and that is the operator. So there are repressor proteins that bind to the operator and prevents the movement of the RNA polymerase and in a way prevents the transcription and translation of the these proteins that code for enzymes that metabolizes the lactose. Now, what happens is that when glucose is present in sufficient amount, the repressor binds to the operator and this particular operon is inactivated. However, when glucose is less and lactose is present, so lactose binds to the repressor and inactivates the repressor, telling the operon, see, I am present now. You please transcribe and produce enzymes to metabolize me. So that is the purpose of the, this is how the repressor is prevented to attach to operator, and this is another type of regulation that happens in the operon. So just for revision part, when glucose level is low, CAP is activated and CAP binds to CAP site and it promotes the operon from transcribing. On the other hand, when lactose is present, that also has the similar effect. It will inactivate the repressor and it will cause the transcription of LAC-Z, LAC-Y, LAC-A-G to produce enzymes for metabolization of the lactose. So this is about the turning on of operon. So as I said, when the glucose level is low, the CAMP binds to the cap protein and it promotes the polymerization by it promotes the transcription by allowing the RNA polymerase to sit at the promoter. But when glucose is high, this process needs to be stopped. The lac operon needs to be inactivated. So when glucose level is high, CAMP is not produced, CAP is not activated, and promoter is not, the RNA polymerase is not allowed to sit at the promoter and perform the function. Similarly, when glucose level is high, lactose level is low, then repressor lactose does not bind to the repressor, and repressor prevents the movement of RNA polymerase and activation of this particular operon. Just like the lac operon, there is a tryptophan operon, and the tryptophan operon also has a similar regulation mechanism. So this is a operon, and it has several genes that are required for the biosynthesis of tryptophan. And just like in the previous case, there is a repressor that can bind to the operator and prevent the RNA polymerase from transcribing all these genes. Now, how will the cell give a signal for the transcription of these genes? When tryptophan level is high, the need for biosynthesis is not there, and the transcription of these genes need not occur. So in that case, when tryptophan is high, tryptophan binds to the repressor, and the whole thing will bind to the operator, and it will prevent the transcription of these genes because biosynthesis is not needed. Already there is enough tryptophan. However, when tryptophan is not there in the environment and there is a need for biosynthesis, so the repressor is inactivated, because here in the previous case, lactose in the form of allolactose was binding to the repressor and activating the and was inactivating the operator. Whereas in this case, when the tryptophan is enough present in quite a large amount, the tryptophan binds to the repressor, and actually it activates the repressor and binds to the operator, and it prevents the, so the repressor is functioning differently in both the cases. In lactose case, the binding of allolactose to the repressor is actually preventing the repressor from binding to the operator. Whereas in this case, when tryptophan binds to the repressor, it binds to the operator, and then it prevents the movement of RNA polymerase. Here is the figure that shows that when tryptophan binds to the repressor, it becomes active. It prevents the RNA polymerase, and there is no transcription happening. Whereas when there is no tryptophan, the tryptophan repressor remains inactive and the polymerization happens. So this is how a cluster of genes that come together, that are localized in a region, can influence the regulation of prokaryotic genes. Thank you very much.